transducent is the G alpha, rhodopsin is the GPCR. What happens, so in the null position, we're not getting any signal from water. What happens is then light comes in, right? It converts the cis conformation of rec now to the trans. So, like the light comes in, right? And instead of having that cis conformation, that makes it trans. That's going to shift the conformation, right, of the GPCR. It's going to fluctuate, right? It's a totally different stereochemistry. And what does that do? It causes GDP to leave and intracellular GTP to bond, right? It breaks this connection. What is the function? I know I'm kind of joking. What is the function of the beta gamma converter? What is this? We know what this is now. This is the GTPase. We know that rhodopsin is the GPCR. What's the function of beta and gamma? Yeah, they're GDIs. They're inhibitors, right? So they are guanine nucleotide inhibitors, right? And so what they do is they bind to the G alpha and make sure that the high concentrations of GTP in the cell do not exchange with GDP. Only once the signal comes in and the GPCR can actually modulate its structure, breaking this connection between these two. Okay, so those inhibit until GTP bound. Until GTP exchanges for GTP. So once it exchanges, we get GTP bound, and we have our G alpha, also known as transducent. Its downstream effector is what is called phosphodiesterase, PDE. And it is bound to an inhibitor. Inhibitor meaning a protein inhibitor. Something that binds to it, just like the beta and the gamma bind to the G alpha, it binds to the phosphodiesterase, that's what PDE stands for. Phosphodiesterase. It binds to it until this comes over. Then it's in a new conformation. It has a binding partner that has a more favorable binding interaction than with phosphodiesterase. So when GDP is bound, it's in a conformation that actually provides a lower, more stable interaction with the, and basically pulls it away from the So I'm just going to look at that new conformation here. Right, this is all membrane bound. Active, this pulls away, okay? Pull that away. And PDE, what does it do? It catalyzes this reaction where it takes cyclic GMP, right? And just makes 5' GMP. Okay? Now, how does that, what's that affect you? Over here, right, in your thing, there are channels, right? We talked about ion channels. And the ion channel is only open when cyclic GMP is bound, right? And if I drew out the whole structure of that, of the rod, right? And we talked about how there are these things that allow sodium, right, and calcium to go in, and then there's sodium and calcium coming out, right? And it's keeping this electrical potential at approximately 40. I think it's negative 45 millivolts or something. All right. So this is going on when this is open and bound to cyclic GMP. Okay? When this closes, right? So what does phosphodiesterase do? It starts depleting all the cyclic GMP that's holding this pore open, right? Once it removes that, what happens? Once all this goes away, we get closure of this channel, and then what happens is, once this closes, right? What happens is the electrical signal goes through the synapse. So we have what they call depolarization when those close up. And so that's what it's doing. It's this this effector, right? This is the enzyme that's doing this, is depleting cyclic GMP, making GMP. That simple change, okay, is causing this ion channel to close, and that's leading to an electrical signal being sent down through the rod to the edge to the synapse that will lead to the brain, sending that electrical signal to the brain. Okay? And so that's as much as I wanted you guys. Hopefully I wrote that. So you guys okay? Okay. Is there anything wrong with what I just said? Besides my fumbling of words. I had a bunch of chicken before I came. I tried chicken, and the only time I get to eat it is usually when I'm by myself. My wife doesn't care for that. Let's get back to business. Yes.
Does that release the inhibitor back and allow the
Yes.
so back to
Yes. Eventually, right, this will become inhibited again. There's that process of desensitization. There's a process that will basically remove that, so that, or actually remove this. This will become inhibited later. I don't know the exact mechanism for that, but there is a mechanism for, eventually once depolarization happens, there's what? I know this. I ate too much chicken. That's amazing. Right? GTPase is only on, right? This is only going to keep the inhibitor at bay for how long? However long it holds GTP. Once it hydrolyzes it, what happens? Goes back to the inactive form. Inorganic phosphate leaves. GDP is bound, it will be bound up. So, right, as soon as this does its hydrolysis reaction, we're dead. We go back, we're inactive, and we wait for the signal again. So that's how we, and this is always the same. Whether you're talking about the beta adrenergic receptor, or whether we're talking about like rhodopsin transducin, always this is what's going on. This is activating it only as long as GTP is bound. And remember what it is, it's a clock. It's a molecular clock, a biological clock. And it only stays on so long as GTP is bound. Once it does its reaction and takes off the gamma phosphate, it reverts back to a form, a structure now that gets bound and is inactive towards binding to the inhibitor. Shifts to a state now that's there's not enough complementary interaction to really keep it together, if you will. So we good? So do you have any questions on that? So this is always the case. From here to here, no matter what question I give you on a GPCR, the G alpha always has the same function. The G beta gamma always has the same function. Right? The gamma, what does it really do? It just associates the beta to the membrane. not really, it's in such a tight interaction, they're almost never found apart from one another. It's almost covalent, but it's not covalent, but to such strength that you never find them separated. Okay? So that's what's going on here. Does anybody have any questions about it?
Why is it important to modulate the rate of
Yeah, so we want to modulate the rate, okay, to affect how quickly we respond, right? We don't want the pathway, so a G alpha, it does the hydrolysis of GTP at about 2 to 3%. So 1,001, we would only get 2 or 3 turnovers. But what do we know? Our responses to chemical stimuli are much faster than a second time skill, right? We respond in microseconds. Internally, maybe even quicker than microseconds, okay? And so what modifying this does is allows us to adapt to the signal, turn it on quickly, turn it off quickly. If we want to turn this off quickly, right, what do we have to do? We have to hydrolyze GTP quicker. Okay? So we have what we call gaps, right? GTPase activating proteins. We just went over those very briefly, but what do? They come in and they activate hydrolysis. They interact with the G alpha, making it take on a conformation that's even more reactive, speeds it up. So we take this from being 2 to 3 per second, that's how many turnovers, right? 2 or 3 per second, to 2,000 to 6,000 per second, right? So that's constantly turn off, bind, oh, turn back on. You know, we can modulate how quickly our response is to stimuli by having these activating proteins that come around. And these are probably sent by some signal within the cell saying, hey, we got to speed things up here. I don't want to get into all that. I don't really know what all sends that up there. Okay? But there's probably, because there's a signal, right? There's upregulation of transcription and translation of all kinds of proteins. One of those upregulations might be the expression of gaps to help speed up the whole process because we're getting a lot of stimuli. Anybody have any questions? So gaps help increase that to adjust the response time. So this is a question. What does that affect? So we have this mechanism for affecting our response time, how long the signal's on. We also have some other mechanisms, right? We have inhibitory pathways, right? There are G protein coupled receptors that inhibit, right? That inhibit pathways. We didn't learn about that here, but we did for the beta adrenergic receptor. Remember, there's a stimulatory pathway that gets stimulated by epinephrine, right? And then that causes that pathway to be stimulated. Then there's another GPCR that will inhibit that pathway. You want me to go over that? Or is that not a question? Yeah. Mm-hmm. Yes, there's a remediation process, right, where this gets recycled. Right? There's a inactivation. It's shown there. We didn't go over that. That's not something we need to do. If you guys can get this, that's very, very good. If you understand that there are proteins that can come in, activate that, that's very good. Do you want me to go over the beta adrenergic receptor example? Epinephrine, yeah. So it's just a slightly different. I'm going to leave all this together here. The only difference, right, is the effector. So I'm just going to draw it this way. It has an integral membrane portion, right? The target for the beta adrenergic receptor, right, epinephrine activated system. Okay, we have a signal epinephrine, so a molecule, we call it. Comes in, binds. We have this same event occurring, right? Exchange. We looked in detail how that works, right? You don't have to remember the details, but the new effector for the GTPase, right? The G alpha is adenocyclase, okay? There are two subunits on the intracellular side, on the cytosolic side, right? There's an active domain or a catalytic domain, and then there is a domain that is regulatory. In your book, they like to give that side a green color, and they like to give this side a red color, right? At least in the figures that I put up. Now what happens, right, same thing. Epinephrine binds to the receptor, right? We talked about how there's a structural transformation, right? This is what we call, again, a guanine exchange factor. That is what the GPCR is. It causes guanine exchange, right? Guanine nucleotide exchange, activating the G alpha. Now it comes, interacts with adenylate cyclase or adenyl cyclase, right? And it takes it from an inactive form to an active form that takes ATP, adenosine triphosphate, and makes cyclic AMP. This is known as the secondary messenger, right? Okay. Cyclic AMP comes, and there is a protein kinase A. It's inactive when there is no, so iron is inactive. When no cyclic AMP is back around, this is inactive, this protein kinase A. What do kinases do? Phosphorylate other proteins, right? That's the enzymes that phosphorylate other proteins. When it binds to it, and that's what I'm showing here, it's coming interacting with this, then that binding event changes its structure from being inactive to active, and now it goes, and then there's a cascade of kinase activity that leads to a cell response. You don't have to go through all the different things, but that's what happens, right? We make a secondary messenger that activates protein kinase A, then protein kinase A becomes activated and it goes and it phosphorylates a number of other enzymes that upregulate the signal. And ultimately it goes into the nucleus and we release what? Glucose, I believe, through this pathway. And I showed you that in the newest version of the what? What? Of the, man, where's the, all right, of the, this is how you guys feel on my exam, right?
Yes, that's exactly how I
Of the presentation, right? Of my presentation. I threw in a slide that basically showed how one signal here would make a response that yielded 10,000 glucose molecules to be let out, right? So we see these cascade reactions where one molecule activates an enzyme. This activates about 100 molecules, right? It's 100 molecules. That activates, each one of those activate 100 of these. 100 of those go and activate 1,000 other proteins, and we get this cascade event that amplifies the signal. So in the end, we get 10,000 molecule signal in the very end of that cascade. Yeah. You see it right there. Yeah, helping to release glucose from glycogen. Glycogen is the storage molecule. It's a kinase that's activating the components that degrade glycogen. All right, so now that I've figured out my words, all right, how is that? Is that okay? I have not been drinking with this. I've been eating chicken, okay? I only get to eat it every once in a while. See what happens. Yes, so how does this get shut down? One way to kill the signal is get rid of cyclic AMP. There's a phosphodiesterase, PDE. This is different than the one in transducin, in the transducin rhodopsin pathway. This is a different phosphodiesterase. This is targeting cyclic AMP. In the vision pathway, what is the phosphodiesterase target? Cyclic GMP, right? That phosphodiesterase has a different substrate, so it's a different phosphodiesterase. So that's one way to kill the signal. The other way is we have another GPCR. 1, 2, 3, 4, 5, 6, 7. GPCR, and this is an inhibitory pathway. So we have a G alpha that the book likes to call inhibitory, right? And it's bound to its beta gamma, right? And then if we get a signal, okay, we've got one signal, we can't remove that signal because it's already in the bloodstream. How do we knock out the signal? Well, we have another pathway. So we send a new hormone that is responsible for doing the same thing. We've got GTP, right? GP leaves, right? And then what happens? That alpha, that's not the alpha, right? Now with GTP bound, comes over, binds to the regulatory side of the domain, causing a structural rearrangement that even though this wants to be interacting with it, it slows it down or disconnects it basically, turns it into an off structure. So that's another pathway. There's also some other things. There are regulatory domains that we looked at ever so briefly, but things like calcium. Calcium can come in and bind to this regulatory domain and inactivate it as well. Calcium is a signaling molecule. When we have these signals, it's something that's released. And so high levels of calcium, this can sense and it can turn it off. So those are the basic things. You can have destruction of the secondary messenger, right? Have an enzyme, phosphodiesterase, knocks out that secondary messenger, shuts down the pathway here. We can have the signaling from the body or from other cells, hormones that come in and tell it to knock down that pathway. So they stimulate an inhibitory G alpha, right? Or we can have these proteins that come in, right? Gaps that come in and help speed up that reaction and help shut it down quicker, too. To fine-tune that time. Calcium, there's a what we call a calmodulin domain. Most people don't. But there's a domain on the regulatory domain of this. Calcium will bind to it, causing a structural rearrangement that affects the thyroid and shutting it down. So once you guys really need to understand, I know there's a lot of pathways. Those are the three that I want you to know. There are a lot, right? The cell is just so finely tuned. It has multiple layers of regulation so that, if one method doesn't do the job, you have other avenues to help to knock it down, okay? So it's not like 1, 2, that's it. There are other things, and it's very complex, but we can understand it from this basic concept of, we got things that will shut it down, molecules can come and bind to this regulatory domain, and then we have proteins that will help activate the hydrolysis of GDP that will help, shut down or tune that signal. So this is all a way of tuning and fine-tuning the signal to give a really fine-tuned response. Because just by looking at it, does it seem like, vision, does it really look like that's all there is one enzyme performing all this stuff? There's a lot of other things going on that we can't study about it. But this is the basics for how it all works. How that all works with everything around it, that's a difficult one.
What about the insulin receptor pathway?
I didn't say we didn't go over that. I focused on the epithelial growth factor pathway for the receptor tyrosine kinases. The insulin receptor pathway is a lot like the epithelial growth factor receptor pathway. I'll go over that now. So you guys good with GPCRs, right? You guys remember that the beta has a beta propeller? You guys remember that tree structure? That's not the young exam, but
It's only a gram.
it makes learning wonderful. Pretty television. Things like that, right? All right. All right. So you guys get the trimeric G protein receptor pathways, right? Are we doing okay, Kristen? No. All right. We're going to get serious now. I'll tell you about the fried chicken. Okay. All right, so receptor tyrosine kinase pathways. Okay. Remember, what is it that all of them have in common? I showed you a slide where we said every single one of them has an alpha helical transmembrane portion, right? Then on the outside is the binding domain. It binds to its specific, whatever its specific stimuli is. So if it's the insulin receptor, it binds to insulin. If it's the epithelial growth factor receptor, it binds to epithelial growth factor. Okay? So there's some binding domain on the outside that is different for all the different kinds of receptors. That's really what differentiates them. Now, all of them have some sort of transmembrane helical structure that's integral to the membrane. Now, on the inside, all of them have a kinase domain, right? That is what binds them all together. That's what makes them all similar. So basically, this all stays the same, and then there's something unique on the outside that binds to its specific binding partner, to its whatever stimulates it. Okay? So what happens, right? On the kinase domains, there are tyrosines, right? So there's our Y. Remember, Y is tyrosine. Okay. So what happens? We have some stimuli that comes in. Let's look at, well, let's just look at the general case first, and then we'll go into the epithelial growth factor. Right? So stimuli comes in, binds to the receptor. They start out in the monomer form, except for insulin. It starts out as a dimer, okay? So let's just stick with this. Start out as a monomer. Binding of this hormone to the receptor causes dimerization. So it changes its conformation so that now it will interact with its partner. with its partner receptor. So now we've got these bound in there, and what does that do? It brings these kinase domains together, right? With the tyrosine, so we've got Y, push right up, okay. We got this. All right, so what happens? Once they brought into close contact, there is autocatalysis, cross-catalysis, where these domains take ATP, okay, make ADP, and they transfer that gamma phosphate to those tyrosines. So if we want to draw this out a little further, they do that. Step 1, step 2, I think of it that way. And then what happens are these domains are now phosphorylated. So I'm just putting the OPO3 minus, right? That's what happens to the tyrosine, right? Because this is what we're doing. This is our tyrosine group, our group, and what's happening is, you get that. So that's what's happening. We have these tyrosine groups, now they're all getting phosphorylated, right? And then what does this do? This provides a new binding surface for proteins that have an SH2 domain. Okay, what is an SH2 domain? An SH2 domain is a domain on proteins that acts as an adapter and binds to phosphorylated tyrosines, or binds to segments of protein that are heavily phosphorylated at tyrosine residues. So what happens is these proteins come in, right, and bind to it, okay? So this is what we call an adapter protein. It contains SH2 domains. That's what's binding to that, okay? And then what do these do? These activate some effector. So activate. So interaction with this activates that. Okay. That's it in a nutshell. Then this effector goes on and causes a signal. Right? So the effector will activate is an enzyme. Enzyme makes a lot of activated molecules, and eventually that leads to signal. Okay, so let's look at the epithelial growth factor pathway, because that's the one I focus on. The reason insulin receptor is very, very important, especially, diabetes and all that kind of stuff. I focus though this time on the cancer graph, because we can really pick out and understand how cancer is formed by just one mutation in one protein, such as RAS, right? So we good with this? The general, how it all happens? 1, 2, 3, and is this helpful so far? Okay, so let's see how epithelial growth factor receptor. Okay, so same deal. Have a question on the exam. You want to draw out a structure maybe to help you, because that I can sometimes I don't understand what you write, but I can really understand a picture real well. I'm kind of, like a child. Oh, picture things. I like those. Right? So here's, I'm just going to use K for kinase domain, right? So we have here, the signal comes in. So what is this called? This is called ERB2, right? right? Or what these are called, A2. Or 2. That is the epithelial growth factor receptor. So you could all call that epithelial growth factor receptor. That's the specific name in the picture. If you just know epithelial growth factor receptor, I'll be good with that. Okay? Okay, we're good. All right, so epithelial growth factor. We'll just call that EF, epithelial growth factor, comes in, binds to these receptors, right? So step 1, we get the signal. What happens? Step 2, the binding causes dimerization. So I'm just going to draw this as our signal coming in. We get dimerization. That's always the same no matter what receptor tyrosine kinase it is. This brings these into close proximity to one another, right? And so we have tyrosines. I'll just put one. Brings them into close proximity, and then what do? There's auto cross-catalysis, right? This domain is catalyzing phosphorylation on this domain is catalyzing phosphorylation of tyrosis on that domain. So I'm just going to say P, for these are now phosphorylated, okay? Is that okay with you guys? I'll just say PP, 5 PP. Okay, you said something about that? Okay, so now it's phosphorylated. What does this do? Causes recruitment. There are 2 proteins in particular that it recruits. There is GRB2, which is found in very close association with a protein called SAS. I don't want to go into the name of this. Basically, the name is totally irrelevant to what's going on here. SAS stands for son of sevenness. It's from a gene. We don't need to bring it. Okay? So what happens? This causes recruitment. GRB2. Okay. Kinase domains, we got PP, right? And now we've got this GRB2 interacting with sauce, so that is now brought in, associates with the receptor, the kinase domain of the receptor, where it's phosphorylated. Without those phosphate groups, does not have the correct conformation, does not have the correct chemical surface for interaction. Now, once these bind, there is recruitment of RAS. Okay? RAS is a GTPase. RAS is a GTPase, analogous to the G-alpha. So if I were to give a question on the exam, I typically say, compare and contrast, the receptor tyrosine kinase pathway with the trimeric G protein pathway. What's the similarity between all of them? All of them have a GTPase. Okay? All of them have a GTPase. In one case, it's the G-alpha. In the other case, it's RAS. Okay, so what happens when it binds, right? It has GDP bound initially, right? But SAS and GRB, the complex, what do they act as? As a guanine exchange factor. Again, they act in the same way that the GPCR acts. So you see the comparison there? The GPCR now has been replaced by this complex that has to associate with the receptor. So, GDP leaves. Now I want to be clear on this. I've seen this a lot. Remember, that this is not a reaction, this is an exchange. GTP from in the cell, in the cytosol, is exchanging places with GDP. There's no reaction going on there, okay? It's just an opening and exchange. Concentration of GTP is higher inside the cell than GDP. So it will freely exchange. Right? This will diffuse out and that will be replaced. Don't get that mixed up on the exam or if you ever come across that. It's not a reaction, it is an exchange, a displacement. Okay, so once that happens, what happens now? RAS, right, goes and it activates RAF, right? So here's our RAS, GTP bound, and what does it interact with? RAF. RAF1. Okay, RAF is a kinase. Okay? And this complex, it activates it so that now RAS with GTP bound interacts with RAF. Right? And now RAF can phosphorylate MEK. MEK is another kinase. That's what the K stands for. It's a kinase. So this gets phosphorylated by RAF. Then MEK interacts with MAK kinase. M-A-P-K. Kinase. Okay? And so we have MEK, RAF, and RAS, right? All these interactions to activate that, activate the MAK kinase. Then we have the nucleus. When MAK is activated, it becomes phosphorylated. So all these get phosphorylated, or not RAF. MAK gets phosphorylated, MEK, MEK gets phosphorylated, MAK gets phosphorylated. Now, once that gets phosphorylated, MAK kinase gets phosphorylated now. It's not associating with it the whole time. I'm just showing how it's coming in, interacting and phosphorylated. And once it's phosphorylated, it can then be transported into the nucleus. Before that, it's trapped outside and it's interacting. Once it gets phosphorylated, it is transported into the nucleus, and there are transcription factors, June and FOX. So these are what's called transcription factors. Anything about what's the share?
Do we need to know all the way to June Fox?
Yeah, all the way to June FOX.
Did you do this in class? Or just this?
It was on a slide. So I would know it. So, are there any med students here? Veterinary students? Holy cow! You're gonna have to know way more. This is like scratching the
This the intent words.
What's that? I mean. Anybody here want to teach English? Go. After you're done with this, but I'm sure in biology, you memorize all kinds of things.
No, not the risk.
But it's not that bad, okay? All I'm showing is what's interacting with it. Okay. So it gets in there, we have transcription factors. These get phosphorylated, so MAK kinase phosphorylates them. Okay? And what does that lead to? Cell response, cell proliferation. Cell proliferation. Okay? Cell proliferation.
Doesn't that actually lead to
Growth factor pathway that I'm talking about leads to cell proliferation.
That's not gonna change. Cell phase.
So, okay. Somebody just, all right, we're going to, this is the punchline. All right, okay. So, does everybody get all this part, same for every single receptor carries? This part is unique. I could have memorized the insulin receptor pathway. This is the one I want you to know. Yes. MEK is a kinase. It's a cascade reaction. RAF activates MEK. Okay, I should write it this way. Then MEK gets phosphorylated by RAF. Phosphorylated MEK activates MAP kinase by phosphorylating MEK. So it's not like it's 1 to 1 to 1. We're activating enzymes here. If we activate one RAF, it's going to probably activate anywhere from 10 to 100 MEKs. Then they start acting as catalysts, and they can catalyze probably 10 to 100 MEKs. So you see how the signal is amplified? If we start out with one signal here, we get one here, this activates, say, 10, okay? So we went from 1 to 10, and then that 10 can activate 10, or 10 MAP kinases each, right? That's 100, right? And then 100 of those go in and start activating, lots and lots of transcription factors. You can see how it all gets amplified. They are transcription factors in the nucleus. They are inactive until they're phosphorylated. Once they get phosphorylated by MAP, then they regulate, you know what transcription factors do? They regulate transcription and translation of gene targets within the nucleus. Okay, so you get some. So here's a question. Are you leaving right now? You want to hear? You don't want to hear that? You want to hear the punchline? Okay. Somebody's confirming. So what kind of questions can we ask about this? This is important. This is what I went over in class. What is uncontrolled cell proliferation? Cancer. So everybody knows that cancer, right? Cancer. Where does it come from? It comes from a breakdown in this machinery, in this pathway. We specifically looked at one. We said RAS. What happens if we get a mutation in RAS that keeps it so that it does not catalyze the GTPase activity? So, right? If we go here, right? PI, right? Remember, the RAS is a GTPase. It's going to shut down this pathway as soon as it catalyzes the hydrolysis of that gamma phosphate. Okay? What happens if you get a mutation in this enzyme that renders it unable to do its catalysis? There's a high amount of GTP in the cell, right? Higher than GDP. You get a signal. RAS exchanges GTP for GDP. It's activated. But RAS does not have the ability to hydrolyze that gamma phosphate. What happens to the signal? It's going. And it's going to go on and on and on. Hopefully your cell will pick up on this, or your body, and it will say, it's time for apoptosis. This cell's screwing up. Okay? But oftentimes, what happens in 30% of cancer, there is a mutation in RAS. 30%. Single mutation, maybe 2 or 3 mutations. Single amino acids. Okay? One amino acid that screws up this thing, and it will stay on and on and on and on. If your machinery doesn't take care of it, then you get cell proliferation and you get cancer. So could you explain it? What if I said, what happens in the epithelial growth factor hormone receptor pathway if the receptor were to somehow come up with a mutation that led it to always be known? You would get So if you understand these different components of this, you can understand where things like cancer lies. So what I went over in class was, okay, why do you care to learn about immunity stuff? Why are you here to learn about immunity? No, it's to get a grade, get through, I know that. But we learn knowledge so that we can apply it to solve problems. We have to learn how something works right, and then when we see something happening that's wrong, like a mutation of RAS, then we have to be able to go and say, how do we address this problem with what we know about biochemistry? How do we try to right what's going wrong in the pathway? And that's why we learn this stuff. It's not for the sake of knowing knowledge, for the sake of knowing knowledge. It's learning knowledge so that we can apply it to those practical applications. It has nothing to do with the letter grade you get in the final, although I know that is very important. But this is why we learn this stuff, so that somewhere down the line, we may not be working with this pathway or any pathway, but if you're a doctor, say, you might discover that there's a pathway that's screwing up. And it's a lot like RAS, okay? It has a GTPase, and then you can learn something from what you already know and apply it to what the problem is at hand. Got really quiet. Yeah, me kind of have some s'mores and stuff. Sing some songs. What? So RAF is a kind of active phosphorylate. So we don't have to keep on. We don't keep on stuff. That means it's phosphorylate. So RAF gets PP'd on. That means it's phosphorylated. Once the neck gets phosphorylated, it's activated. It goes and PP's on mass. That means it gets activated, goes ahead, so I'll show it like that. Once it's PP'd on, it goes and PP's on. Yeah, RAS this is the effect. RAS is changing the conformation of RAF. And that's the thing that's going to help send the secondary messenger, which is another protein that's going to start. And this is only going to phosphorylate enough next until, right, this RAS associate. That should probably not here. Does that make sense? This is, if you look at the chapter 12, this is very minimal compared to the chapter. But what I think is this is important because so many people know somebody who has cancer, right? And what's amazing is all of it can be attributed to a molecular problem. Mutation or two mutations in one protein can lead to this problem that we see and a lot of people experience where they have horrific death because of the simple molecular mutation. It's pretty amazing. And so, what a lot of people are trying to do is how do we get around it? Can we target RAS? Some people are trying to do what? Gene therapy. Can we just cut off the gene, put in a new gene?
Actually, there's a study on it.
Yeah.
I was reading about that. I didn't read about it because I read it last week.
Yeah. See, and it's not that you have to target this pathway, but by knowing about this, you can start to say, okay, here's something we can address. Here's something we can try. And what we're trying to do is not focus on one answer to the problem, but by understanding this, we can at least start to try to answer the problem. It's pretty amazing. Yeah. You gonna watch YouTube? Yeah. In RAF, activates MEK. Yeah, so that's what I'm showing here. So let me draw it like this. MEK, unphosphorylated, once that binds to it, it gets phosphorylated. The phosphorylated MEK then goes, activates MAP kinase. I'm sorry about that. MAP kinase gets phosphorylated, enters the nucleus, and it has a good time with a bunch of partners in there. They go and have babies. Right? Gene phos. MIC is another one. Do I care if you know every single transcription that you know? MIC, gene phos, those are transcription factors. And they are inactive until they're phosphorylated. So they're just floating around in there, right?
Are they phosphorylated right now?
If MAP gets onto them, yes. So I'll put they come in, they get phosphorylated. There's nothing. I don't know exactly how many. All that I care about is getting phosphorylation. And what does phosphorylation do? It changes its conformation, right? And that new active conformation goes in. I will go into more detail. So is everybody okay with that? All right. Yeah, so so you could think about this in several ways. There has to be a residue, right, that's responsible for breaking that chemophosphate. Do you imagine that? So, I don't know what the exact residue is. Let's just say it's a serine, right? Serine, right? And we've got our chemophosphate here. Right? So let's say it's a serine. I don't care what it is, it's a serine. Okay? What happens? We can have this, we can have a tap here, right? Or what's the tap? Let's make it different. Let's look at the general base, okay? And we have water that comes in. That, attach that, okay? And then let's have it come here, and that goes off there, okay? And we'll get the release of the inorganic phosphorus. That's what PI is. Now, what happens if you get ATP bound and then there was a mutation from a base to, so what's a base? A good base is something like lysine, right? Let's say you have the mutation from a lysine now to a glycine. What's glycine going to do to water? It's not going to act as a base. And so what do you have? You have a perpetually bound ATP. There's nothing there to do it. Right? Does that make sense? How the picture goes? How mutation can change it? You get a complete inactivation of the important amino acid residue just gets completely gone. What's that? All right. So are we good? Yeah. Okay. All right, guys, some of you look like you're dealing with it. Not that bad, we're doing it. All right. How are we doing? All right. Is that done on that top? That's not exciting. Yeah, that's not included. chapter 10 is not directly included, guys, like, I'm not gonna ask you the exact structure. All right, let's